Welcome to my YouTube channel, Big Ideas Simplified. Today, we will be talking about reproductive cloning. If you like my video and learned something new, make sure to like and subscribe to my channel below. Leave me a comment if you have a question and I will respond as soon as I can. Today, I'm talking about a topic that's considered both the dawn of a new era and the plot of a sci-fi nightmare, reproductive cloning. We're going to explore the science, the history, the potential, and the pitfalls of this biotechnology. So, buckle up, because I'm about to make your DNA do a double take. First, let's clarify how cloning works. Cloning involves making an identical copy of an entire multicellular organism through natural or artificial means, resulting in organisms or cells with identical DNA. In sexual reproduction, a haploid egg and sperm cell fuse to form a diploid zygote containing genetic material for a new individual. Additionally, early embryonic development relies on the cytoplasmic material within the egg cell. This idea forms the basis for reproductive cloning. If the haploid nucleus of an egg cell is replaced with the diploid nucleus from the cell of any individual of the same species, called a donor, it will become a zygote that is genetically identical to the donor, producing a clone. Now, let's take a trip through cloning history. Our journey begins in the 19th century, when scientists first attempted cloning. In 1885, biologist Hans Drake separated the blastomeres of two-cell sea-urchin embryos, marking the first time that scientists separated embryonic cells in the early stages of development. In the early 1900s, embryologist Hans Spemann used similar methods to clone salamanders, but only up to a certain point in the original embryo's development. Then in the 1950s, biologist John Gurdon was the first to successfully clone an animal, transplanting the nucleus of a tadpole intestinal cell into an enucleated frog egg. Fast forward to 1996, and we have the grand finale. Biologists Ian Wilmot and Keith Campbell cloned the first mammal. They transferred a nucleus from an adult sheep's udder cells into an enucleated egg, producing an embryo that was carried to birth in a surrogate mother. The cloned lamb was named Dolly. Imagine a world where endangered species aren't at risk of extinction, where abundant agriculture solves world hunger, where organ shortages are a thing of the past, and where infertile and same-sex couples can still have children. This world can be achieved through reproductive cloning. Cloning can result in the successful revival of extinct species. By using the genetic material of dead organisms, cloning can expand gene pool diversity. Cloning can also allow scientists to study the extinct species as living organisms. Cloning can be used to increase agricultural production, particularly livestock and fresh produce. This is because production would be faster than the natural process, and the number of organisms produced at a given time would also increase. Doctors could use cloned tissue when needed for surgery on the original organ or for organ transplants, allowing for more patients to be treated and avoiding wait lists. Children could be produced through cloning without donor eggs or donor sperm, so couples would only need a surrogate mother to carry the clone until birth. Of course, no technological breakthrough comes without its cautions. Enter the dark side of cloning: genetic defects, ethical issues, unpredictability, and timing. If the original organism has genetic defects, these transfer to the clone. For example, Dolly the sheep only lived 6 years because of tumors in her lungs, which may have been in the genome of the original sheep Dolly was cloned from. Critics say that clone production requires the exploitation of life. In the case of Dolly, it took more than 277 attempts before a viable clone was produced. This suggests that if humans were to be cloned, scientists would need to conduct numerous failed tries. Most clones that have been produced so far have had reduced lifespans and have developed health problems. Also, even though clones are genetically identical, their behaviors might be different depending on the environment and nutrition. Especially considering the conservation of species, the process of cloning endangered species has proven to be more difficult than other domestic species. It will likely take decades of additional research time and money to achieve. In conclusion, from sea urchins to sheep, the journey of reproductive cloning has been a roller coaster of discovery, with twists, turns, and more than a few ethical speed bumps along the way. While scientists will need to proceed with caution, whether you're team clone or team original recipe, one thing's for sure. The future is cloning, and it's anything but copy-paste. Thanks for watching. Stay tuned for more big ideas, simplified content.